So I'm going to present you my thesis called Environmental Preferences and Spatiotemporal Distribution of the Main Genera of Phytoplankton Present in the Bay of La Paz, which I undertook at the CIBNO here in La Paz, Mexico, under the supervision of Raúl Martínez Rincón and Miguel Matus Hernandez. So first, as an introduction, phytoplankton is a very important component in the marine environment since it is located at the base of the marine food webs. It is responsible for roughly 50% of the primary production in the ocean and acts as a climate regulator. However, the distribution of phytoplankton depends on light and nutrients, factors that are influenced by environmental variables such as temperature, salinity, pH, etc. Phytoplankton is known to be sensitive even to small environmental changes, which lead to questions such as which variables regulate phytoplankton distribution and how, questions that could give answers about water quality, help to enhance food production, and to prepare against red tide events or harmful algae blooms, to understand their adaptive capacities in changing environments and their ecological and biogeochemical functions. So my thesis was based in the coastal areas, and coastal areas are subject to high anthropogenic impact, which makes the monitoring in those areas very important. However, the conventional methods are often time-consuming, geographically incomplete, and expensive. That's why in the last years, researchers try to focus on other techniques, such as remote sensing. However, the satellites like seaweaves and MODIS have a low resolution, and since coastal areas are complex environments, the Landsat 8 satellite with a resolution of 30 meters is an ideal tool. The main objective of my thesis was to determine the environmental preferences of the main phytoplankton genera in the Bay of La Paz, and to analyze their influence on distribution, composition, and diversity by means of species distribution models. So first, the methods, my study area, was located in the south of the Baja California Peninsula, and more specifically, in the southeast of the Bay de La Paz. So here you have my study area, where I wrote down the most important features that are going to be recurrent in my results. So up here we have the San Lorenzo Channel. This area here, I called it the offshore area. This here is the Ensenada de La Paz, which is linked to the Bay de La Paz via the channel. And where the channel meets the Bay de La Paz, this is called the transition zone. So a bit more about data acquisition. The data was previously acquired between August 2016 and July 2017. In that period of time, the phytoplankton data was obtained by means of trawls with 20 micrometer mesh. The samples were then preserved in Lugol and later on identified under the microscope to the lowest taxonomic level possible. Then the relative abundance was calculated in order to have the data in number of cells per milliliter. At the same time, the environmental data was collected using a multi-parameter sensor, and variables such as temperature, dissolved oxygen, chlorophyll A, pH, and salinity were measured. The data was synchronized with the Landsat 8 overpass. So a bit more about the Landsat imagery. So the Landsat images used in this thesis were downloaded from the USGS Earth Explorer server between the years 2013 and 2018. The images were then processed with the raster package in R in order to calculate the reflectance. The reflectance was then combined with the in situ measured variables, and then the best fitted multiple regression models developed by Erwin Hernandez in 2018 were applied in order to predict the environmental factors for this period of time. So now about the statistical modeling. So from the 38 genera we identified in this study, only 12 were selected based on their frequency of occurrence. So all the 12 genera selected were at least occurring 30% of the time in our study. We also selected our variables based on collinearity, and we observed that temperature was correlated with dissolved oxygen as well as with pH, and so we decided to remove temperature from our variables. Here you have the general function formula of our generalized additive model, for which we used the Poisson family because our data was integer and positive, log as link function, and the level of smoothing was equal to 4 in order to avoid overfitting. Then we applied the forward stepwise selection based on the lowest AIC in order to obtain the best fitted model for each genus. From those best fitted models, we were able to obtain our effect plots to analyze the effect of the variables on the phytoplankton abundance. And combining the best fitted model with the seasonally averaged variables from Landsat, we were able to predict the phytoplankton distribution seasonally. Lastly, about the diversity. So for the diversity, we analyzed the abundance of all 84 identified species in that case with the Shannon-Weaver index using this formula here. And then we applied a generalized additive model following the same methodology as described before in order to analyze the effect of the variables on the species diversity. Now the main results and the discussion part of my thesis. So first, we were able to identify autumn as the season with the lowest number of identified genera and spring with the season of the highest number of identified genera. In this graph, the genus that was the most abundant throughout the study was Chaetoceros. Here you have a list of all the best fitted models that we obtained for our 12 genera, with Pleurosigma being having the lowest explained divines of 16.2 and Thalassonema the highest explained divines of 80.2. So now the environmental preferences and seasonal predictions, first for Tripos and Dinophysis. So both genera showed a preference for low chlorophyll A. They were predicted to occur mainly in the offshore area during winter and to be absent in summer. So this is due to the fact that so they occur mainly in the offshore area because the area is nutrient poor and deeper than the rest of our study area. Then the reason that Tripos is absent in summer is due to the fact that it avoids the surface layers at high irradiance, as it has been shown in literature. And the irradiance is the highest during summer here in the Bay of La Paz and the lowest in winter, which explains this high abundance in winter. For Dinophysis, however, it showed a higher affinity for deeper and stable waters. Deeper waters, so again, coherent with our results in the offshore area where the area is deeper. And stable waters because in winter the water masses here in the Bay of La Paz are more stable, whereas in summer they are more mixed. Chaetoceros showed a preference for low salinity and low dissolved oxygen and was predicted to occur mainly in the offshore area during summer. In the season of summer, the dissolved oxygen is lower in our study area since oxygen dissolves less good in warmer temperatures. Also, this genus was associated with high temperatures in literature, but this leads to the question, why doesn't it occur in that case in the Ensenada where the temperatures are the highest? So this is due to the fact that the salinity increases strongly during summer due to the high evaporation rate, and a reduction of growth of Chaetoceros has been observed at high salinity by Rayal in 2014. So this might be an explanation why it doesn't occur in the Ensenada. Now, Prorocentrum, also affine to low dissolved oxygen, was predicted to be highly abundant in the offshore area in summer and was absent in winter. So this genus, as well as Chaetoceros, is also associated with high elevated temperatures. And again, same question, why doesn't it occur in the Ensenada? This is due to the fact that species such as Prorocentrum lima it has been observed that there is a growth inhibition at temperatures above 30 degrees, temperatures which are definitely reached in the Ensenada during summer. Now, Eucampia Navicula, who are very interesting genera because they present completely opposite environmental preferences. Eucampia prefers high chlorophyll A and Navicula low chlorophyll A. They both followed similar distribution patterns, as they were predicted to occur in the Ensenada only in winter and autumn, and along the coast and the transition zone in summer. So this is due to the fact that the Ensenada presents a high amount of chlorophyll A, but also during spring and summer the temperatures, as I said before, increase strongly. And both genera are diatoms, which are known to prefer cold, nutrient-rich and dynamic waters. So there might be an explanation that they occur in the Ensenada since the chlorophyll A is high, but when the temperature increases, they leave the Ensenada to be more abundant in the offshore area. The distribution that is limited in the coastal area in summer is probably due to the fact that in this area the chlorophyll A is high, but this area is less warm than the Ensenada. Now, Gyrosigma, Pseudonychia, and Thalassonema, who all preferred high salinity and intermediate chlorophyll A, they were all limited to the Ensenada and the Channel, but the highest abundance of Gyrosigma was predicted to occur in winter, whereas for Thalassonema and Pseudonychia, this was more the fact in summer. So all the three genera are related to areas of high nutrient input in literature. They were associated with upwelling areas or river runoffs. The reason that Gyrosigma was mostly abundant in winter is due to the fact that this genus is mostly related with cold temperatures, whereas Pseudonychia and Thalassonema are very tolerant to high salinity and high temperature, which makes them ideally adapted to the environment in the Ensenada in summer. Now, Pleurosigma and Rhizosolenia. Pleurosigma showed a preference for intermediate chlorophyll A, and Rhizosolenia was especially preferred high salinity. So both genera were present the whole year long in the entire study area, but the highest abundance of both was predicted to occur in the Ensenada and the transition zone during the summer. So both genera are tolerant to a wide temperature and salinity range, which explains the wide distribution in our study area. But the highest abundance in the Ensenada during summer is due to the fact that in this area the chlorophyll is high, as well as the salinity. Protoperidinium was also present year-round, but was predicted to occur mainly in the offshore area during spring and autumn, and showed a preference for low chlorophyll A and high pH. So why in the offshore area? Because in this area the chlorophyll A is less concentrated. The decrease of abundance that is observed in summer is due to the fact that the pH is decreasing and the chlorophyll A is increasing, so it doesn't meet the environmental preferences of this genus anymore. Also, this genus is very well adapted to the nutrient-depleted environment after the summer, because it is a motile dinoflagellate which is able to migrate between the nutrient-poor surface layer and the nutrient-rich bottom layer. Also, this genus has been shown to survive and even to resume growth after 20 to 70 days of starvation, so again, very well adapted to nutrient-poor environment. Lastly, the diversity. So the area with the highest species diversity has been identified as the transition area and the channel. The lowest diversity was observed in the south of the Ensenada, and the season that was the most diverse was summer. So first, why is the diversity so low in the south of the Ensenada? This is due to the fact that the south of the Ensenada is actually a muddy flat, so logically there are less phytoplankton occurring in this area. Then the channel and in the transition area, the diversity is so high since both are very shallow areas where important currents are occurring, which are actually resuspending the nutrients in the water column. And since tropical and subtropical phytoplankton is limited by nutrients rather than by light, this could be an explanation why this area is so diverse. And lastly, why is summer the season with the highest diversity? This is probably related to the fact that at the late spring, southeasterly winds bring nutrient-rich tropical surface water to enter the bay via the San Lorenzo Channel up here. And since the San Lorenzo Channel is connected with the Ensenada through currents and tides, they constantly exchange, and so this additional nutrient input could be the reason why summer is so diverse. As a conclusion, we can say that generalized additive models as well as Landsat-derived environmental factors are very useful tools to describe environmental preferences and diversity of phytoplankton. Community shifts have been observed and the most important variables have been identified. Moreover, the channel and the transition area are most diverse and hence important areas in which management strategies for aquaculture, fisheries, or public health could be applied in the future. Thank you very much.